Now, in viral replication, there are several different steps to replication, and where this becomes really essential is in treatment of the virus. So you understand how the virus replicates within a host cell. You can understand what's unique to viral replication, and therefore what the target might be in antiviral therapy. So let's just look at the replication scheme for viruses, and this is overall a general scheme of replication. When we get into the specifics for DNA viruses and then positive stranded RNA and negative stranded RNA viruses, we will speak to the specifics of each of those viral families. The first thing any virus has to do is attach to a host cell. All right, so that's the most important thing that a virus can do when it gets inside of the host. It has to be able to find its target cell, and it has to be able to attach. If it can't attach, it can't initiate an infection. And that's what antibodies are good for. You make a nice, good antibody response to a virus, or you get vaccinated for a particular virus like influenza. Those antibodies can bind up that virus and prevent this very first stage of viral replication. So, attachment we're showing here with the naked virus attaching to a host cell receptor, but keep in mind whether it's naked or enveloped, all viruses have to start with attachment. Now the very next step is penetration. So the virus, being an obligate intracellular pathogen, once it attaches, has to penetrate that host cell in order to get in to the cytoplasm and eventually be able to replicate. The next step that has to happen with viral replication is that, remember, we have to get our nucleic acid made. So what has to happen next is we have to lose that capsid, and that process is called uncoating. So that's the third step of viral replication, where we're uncoating, releasing the nucleic acid, which is shown here in red, from that viral capsid. The next thing that's going to happen for viral replication is you're going to take that genomic material, whether it's DNA or RNA, and you're going to begin to replicate it. Now, DNA viruses, and we'll talk again more specifically when we talk about the family, but DNA viruses, because they're using all of your host cell machinery, DNA viruses have to travel to the nucleus to replicate, because that's where you replicate your DNA. So that's where it has to replicate its DNA. And the only exception to that rule is pox virus, that takes everything that it needs with it. Okay? So we're going to replicate our genome. Viruses that are RNA don't have to go to the nucleus because they're RNA viruses. So they actually can replicate within the cytoplasm. So synthesis of the genome, replication of the genome, and making proteins, both early and late proteins, are going to happen at stage 4, which is the macromolecular synthesis. Now, typically, the early proteins that are made go into helping the cell replicate. Any specific enzymes that the virus might need are usually made very early, and then late protein synthesis generally makes the proteins that are required for the house that the virus lives in, the structural house. So we have early protein synthesis, we have late protein synthesis, and any modifications of those proteins that have to happen occur in the post-translational modification. So we have a protein, we need to add a sugar, that's going to happen in stage 5. Now, all of the viruses will have to assemble at some point. So that's the next stage of viral replication, assembly of new virus particles. So all of the proteins that are made are going to hook up with nucleic acid, and they're going to surround that nucleic acid and assemble within the cell. Now, assembly is going to be really helpful for you in terms of diagnosis for viral infections. And the reason that is because, again, we've mentioned that we've moved from a prokaryotic system to this obligate intracellular pathogen. We can't culture it on different types of media, all right? They're probably not going to give you serological data or molecular data. Oh, we did a PCR for herpes and it was positive. No, they're not going to do that. So what we have to rely on a lot of the times are pathology results. So you can actually see in the cells where the virus is assembling a lot of the times, and that translates into something that you can actually detect and use for diagnosis. And a good example of that is a Negri body that happens with rabies virus. The assembly site for the virus in the neuron is detectable and called a Negri body, and that is positive for diagnosis for rabies virus. So that helps you translate that into something that's clinically relevant. Now, the last stage of viral replication is release of the virus. It doesn't want to stay trapped in the host cell. It wants to get out, and it wants to go infect another cell. So release is going to be an important step, and that's going to be our last step. There are two kind of different ways that a virus can leave a cell. If it really wants to make, a big splash, it's just going to replicate to really high titers in the cell and blow up the cell and go on and infect another cell. And a good example of that are the polioviruses. They all grow to really high titers. They just basically capsize all of the host cell protein-making machinery for their cells. So they replicate to really high titers within the host cell, and then the host cell blows up, and they go and infect another cell. Other viruses, like the herpes viruses, for example, a lot of the enveloped viruses, as we're showing here, will push through the host cell membrane, and we call that budding. In that process of budding, the cell is pushing through. If I'm the host cell membrane and here's the virion, it's pushing through and capturing that cell envelope to use as its own envelope. Now, all of the envelope viruses pretty much are going to do that. Some of the envelope viruses, like herpes viruses and the paramyxoviruses, have what we call a fusion protein. And fusion proteins allow a virus to really leave one cell and cause it to fuse to another cell, so it never really has to get outside of the host cell. It can escape immune detection that way. But for you, what you're going to notice with the viruses that cause cell-cell fusion is that, again, we get a little bit of pathology. And what happens is you get the formation of giant multinucleated cells. So I'm a virus. I guess we'll use the Ebola virus as an example here. Here's the giant microbe for the Ebola virus. Let's just say this little guy, we'll ball him up a little bit. He's pushing through a host cell, and now he causes fusion, let's say, to another cell, leaves that cell, causes another cell to fuse together. So basically what you've got is you have one giant cell that is multinucleate, and that is helpful for diagnosing viral infections such as herpes and some of the paramyxoviruses. Now, another bit of terminology that we use is those giant multinucleated cells are also called syncytia. So you want to keep those terms together. As far as antiviral therapy goes, we said that viral replication is clinically relevant for you because you have to know the different stages of viral replication to understand that antiviral therapy. Most of the antiviral drugs target the molecular synthesis. So they're targeting usually replication of the DNA or replication of the RNA. We do have some antiviral drugs that work against the uncoating, and the antiviral drugs for influenza, for example, the amantadine and rimantadine, target uncoating. We actually now, for treatment of HIV, have fusion inhibitors. Okay, so in order to really understand the mechanism of how those antiviral drugs work, it's a good idea to understand viral replication as a whole. All right, so what we want to do now is just kind of go through and talk about some salient points that go with each one of those steps for viral replication. Now, before we begin that, it's important to understand that viruses spread simply by the same mechanisms that most bacteria spread, okay? So we're talking respiratory, sexually transmitted, fecal-oral contamination, all of the same mechanisms that we've talked about for spread of bacteria apply for spread of viruses. Now we do have a whole family, really, of viruses that are called arboviruses. And arbo comes from the phrase arthropod, there's your R, born, bo, arboviruses. These viral families are all spread by an insect vector. And mostly we're really talking about mosquitoes. And the main families that are arboviruses are the togaviruses, the flaviviruses, and the bunyaviruses. So, spread of these organisms or pathogens is similar to what we talked about for bacteria. And in this case, though, we have a lot of insect vectors being used where the insect is not necessarily the tick, but now the mosquito becomes the predominant vector. Mosquitoes, again, just like we mentioned, are the most common vector and mechanism of spread, but you can have other vectors. Ticks are also capable of spreading viruses, and a good example is the Rio virus, Colorado tick fever virus. You can also have other biting arthropods that spread viral infections, but they're much less common. So predominantly, really, we're talking about the mosquito. Now, attachment, we mentioned, was the first step and a very important step in viral replication. And it doesn't matter if your virus has an envelope or if it's naked, the first step has to be attachment. And if you can block attachment, you have then successfully interfered with that viral infection. And the antibody response that we make during a viral infection or when we're vaccinated is designed to, in viral infections, prevent this very first step of viral replication. Now, a couple of other points to understand about the attachment stage of replication is, number one, that it determines the host range of the virus. So by way of the receptors that I can attach to, that determines my host range, i.e., can I bind to humans? Can I infect cats? Can I infect dogs? All of that is determined by your attachment and the different receptors that you're capable of binding to. It also determines tissue specificity. Okay, so am I a virus that can go to the CNS? That is determined by the attachment. And so the different target organs that you're going to see that are affected by a virus have to do with, again, attachment proteins and where the virus can go and what's going to allow it to get in. So what we're showing on the bottom here, again, is simply just that with enveloped viruses, you need a receptor on the host cell, and with naked viruses as well, you have to get in through attachment. So that's a very important step in viral replication. Speaking of viral attachment, there are some receptors that the USMLE expects you to know. Specifically, you should know HIV. Most of us know about CD4. That's the receptor that's found on T helper cells and macrophages and some dendritic cells that allows the virus to attach to those cells. But you also have to know the co-receptors. The co-receptors for HIV are CCR5 on the macrophage and CXCR4 on the T cells. And those have been shown to be essential for viral infection with HIV. We'll talk about them a little bit more when we get to HIV, but you should know in addition to CD4, what the co-receptors are for HIV infection. Now, Epstein-Barr virus, also essential to know that EBV is targeted to B cells because it uses the CD21 molecule as its host cell receptor. So it is targeting the B cell because it expresses that CD21. The rabies virus actually uses the acetylcholine receptor, and that's what helps it get into the neurons. So these are some of the receptors that you really need to have a good idea of. And the last one that's listed there are the rhinoviruses, which clearly target the nasal mucosa, and the majority of them, probably over 85%, actually utilize the ICAM receptor. Okay? So as you're studying some of the stages in viral replication, it's also a very good idea to know what some of the receptors are. Now, naked versus enveloped virus. All right? This is really actually an important point because for a lot of students it seems backwards compared to what it actually looks like. With a naked virus versus an enveloped virus, which virus is more sturdy? Okay? Students always assume that it's the enveloped virus because it looks like it has this whole extra layer of protection. But it's actually the other way around. Okay? Naked viruses are much more sturdy than enveloped viruses, and here's why. The envelope is derived from a host cell membrane. So just as sturdy as your host cells would be after a sneeze and spray on the desk, as soon as the air hit those cells, they would basically dehydrate and shrivel up. The same thing happens with enveloped viruses. They are very sensitive to drying and detergents because the envelope comes from a host cell membrane. Whereas the naked virus, think about this, the naked virus is simply just a piece of nucleic acid surrounded by a protein coat. A very sturdy protein coat. An icosahedron is extremely sturdy. Okay? So when it gets exposed to drying, to detergents, it's extremely hardy. Now, how does that translate into something clinically relevant? It's very important as far as means of transmission. Here, I'll give you a couple of examples. First is that only non-enveloped viruses can use the GI tract as a portal of entry. Why is that? Because if an enveloped virus gets ingested, gets down into the stomach, the acidity of the stomach will chew up the envelope. Okay? And the little virion package that's left does not have the attachment proteins that are necessary to bind to the host cell receptor. So basically, that viral infection has just been eliminated. With a naked virus, you ingest that virus, very sturdy outer structure with encasing nucleic acid. That is the only type of virus that is going to withstand the GI tract as a portal of entry. So, Norwalk virus, which is a major cause of viral gastroenteritis, rotavirus, which is a major cause of infant gastroenteritis, polioviruses, which don't cause gastroenteritis but use the fecal-oral route as their means of transmission, they are all naked viruses. Okay? So that's a very important point as far as the naked viruses are concerned. Enveloped viruses, a lot of the times you'll see, for example, when we talk about the hepatitis viruses, you'll see that the ones that are enveloped are much more sensitive. Those are the ones that typically have to be spread by close contact, sexual contact, or shared needles. Whereas hepatitis A, for example, that's a naked virus. How is that one spread? It's spread through fecal-oral. Okay, now, vaccination strategy for the different types of virions. Antibody is really very helpful for controlling the spread of naked viruses. Okay? So if naked virus gets into a cell, infects a cell, and blows it up, if antibodies there, it can bind to that virus and prevent attachment to another cell. So antibodies very helpful. Now, with enveloped viruses, antibodies are helpful too, but cell-mediated immunity is really required because we're talking about budding from a host cell. We may be talking about moving from cell to cell to cell, where you really need a good cell-mediated response. Now, I'm not saying in general, we're talking really about vaccination strategies here, but overall we know that because viruses are obligate intracellular, that we do need a good cell-mediated immune response. And that translates into if you don't have good cell-mediated immunity, for example, if you have a T-cell deficiency, you are going to be more susceptible to viral infections. All right? So here we're really just speaking to the vaccination strategy for the different virion types. Now, viral entry into host cells, as we talked about a little earlier, can happen by one of two mechanisms. Naked viruses are generally going to attach to the host cell and cause endocytosis of the virion in order to take it up into the host cell. Whereas a lot of the times envelope viruses, because the envelope was originally derived from a host cell membrane, can fuse back with that host cell membrane and just dump the virion into the cytoplasm. So those are a couple of different entry mechanisms that are used for the different types of viruses. Now, macromolecular synthesis, okay? Macromolecular synthesis is something that I know gets a little bit confusing, but if you really think about what's going on, it's really not so bad, I promise, okay? DNA viruses. DNA viruses have to replicate in the nucleus, okay? Remember we said pox was the exception. DNA viruses have to replicate in the nucleus because, remember, viruses are so small. They really can't take all of the enzymes and proteins that are needed to replicate themselves, so they're utilizing the host cell machinery to do that. All right, so the DNA viruses are going to go to the nucleus, okay? Clearly, in order to get proteins made, they're going to have to use what? A messenger RNA, okay? Now, the RNA viruses get a little bit trickier, but really they're not bad, okay? RNA viruses come in two flavors, okay? You have a positive-stranded RNA virus and you have a negative-stranded RNA virus. If you write this down and highlight it, this will be the easiest thing that we talk about. A positive-stranded RNA virus is equivalent to a messenger RNA, okay? A positive-stranded virus is equivalent to a messenger RNA. So the first thing that happens when a positive-stranded virus gets into the cytoplasm of a host cell is it gets what? Translated. And then it makes all of the proteins that it needs to replicate. Now, in order to make genome, it has to go through a negative-stranded RNA template, okay? So we're starting with a positive strand of RNA. We get translated as soon as we get inside a cell. And the next thing that happens is we're making all of the little proteins and enzymes that we need to replicate. One of those is going to be an RNA-dependent RNA polymerase that's going to help us make genomic RNA, okay? And in trying to make our genome, we have to go through a negative-stranded RNA template, okay? Now, the RNA-dependent RNA polymerase is an important thing that the RNA viruses have to either code for, like the positive-stranded viruses. The negative-stranded viruses, in addition to coding for that enzyme, have to bring it with them inside the virion, okay? Now, negative-stranded RNA viruses, okay, again, are a copy of the message. So what has to happen here? What's the first thing you think has to happen in a host cell with a negative stranded virus? You have to get it copied into messenger RNA. So all negative stranded RNA viruses bring with it in the virion the RNA-dependent RNA polymerase. Then it can make the template copy of messenger RNA. That messenger RNA is not only used to make protein, but it's used as the template to make more genomic RNA. All right? Now, retroviruses are kind of the oddball here. Retroviruses are essentially positive stranded RNA, but they certainly don't follow any of the dogmas, okay? So HIV, as a positive stranded RNA virus, uses what as a template? Double-stranded DNA, okay? So it really goes backwards in macromolecular synthesis. And again, when we get to each of the different viral families, we'll talk about this again in a little bit more detail so that you have a good understanding of the different types of genome synthesis with all of the different viral families. Continuing on with the replication of nucleic acid, and again, this is just kind of reviewing some of the things we talked about, and in the next slide we're going to do the same thing. So by the end of this, you should have a really good idea about how these viruses replicate. Now, the first bullet point here is progeny viruses will have a nucleic acid sequence that is identical to the parent, okay? And what we're talking about in this second bullet point that I'm going to really demonstrate probably a little bit better in the next slide is that the single-stranded viruses, like we discussed, let's say for a positive stranded RNA virus, has to use a replicative intermediate that is a template. So for example, if you were in a very simplistic description, if you had a poly U, okay, the replicative intermediate would be the opposite of that. It would be a poly A. So that's what the virus has to go through to make more genome. So then it would look at the poly A to make the parent genome, which is going to be the poly U. This table really describes the replication of genomic nucleic acid very well. If you look at the column that says parental genome, you're going to notice that progeny genome is exactly the same. The parent and the progeny are going to be identical. So really, all you have to do is look at the intermediate replicative form. So let's start with the positive stranded RNAs, okay? The genome is a positive strand of RNA. We said that in order to make more genome, it has to make a copy of itself. So the replicative intermediate is going to be a negative strand of RNA, all right? Then it will use that negative strand to copy and make more parental genome. Skip retroviruses just for a second. Go down for now and look at the negative stranded RNA viruses. It's going to be exactly the opposite, okay? So your parent genome is negative stranded RNA. Your progeny genome is negative stranded RNA. What's in the middle? The template. The template that is going to be the opposite of the parent, which is a positive strand of RNA. Okay? Now you can go back up to retroviruses. I told you that the parent genome for retroviruses is really positive stranded RNA, but it does not use anything close to that replication scheme of the positive stranded RNA viruses. So, progeny genome, again, is going to match the parent. What's unique and different about the retroviruses is that the intermediate that it uses is actually double-stranded DNA. So retroviruses take a positive strand, single strand of RNA, convert it to double-stranded DNA, and then use that double-stranded DNA to make parent genome. Now, skipping down to the last two viruses listed in the table, the double-stranded DNA viruses. DNA does not use a replicative intermediate, right? DNA is made from DNA. So we don't need a replicative intermediate when we're talking about DNA viruses. The exception, though, is hepatitis B. Hepatitis B viruses actually do utilize a reverse transcriptase, but they go from double-stranded DNA, and their intermediate replicative form is actually single-stranded RNA that they then use to generate more double-stranded DNA. So that's a good summary for you of the different types of replication for all of the different classes, really, of the viruses we're going to be talking about. And again, before we get to the major ones, we're going to go through some specifics of viral replication. Now, release of viruses. Again, naked viruses, we said, are going to blow up the host cell. But enveloped viruses, as I'm showing here, will actually push through that cell membrane. So you can kind of see the stages. You can see it pushing through, getting a little bit further, and finally, in this last image, butting completely through the membrane, but taking it with it as its envelope. One of the things I have to point out, though, is you'll see on the cell membrane these little lollipop-looking figures, one with a square and one with a triangle. Those are viral-encoded glycoproteins that are essential, okay, because that's what the virus is going to use to know how to attach to another cell. So those little stick figures there are really important for the virus. If a virus butted through a cell membrane without those little viral-encoded glycoproteins, it would be non-infectious. It would not be capable of binding to another host cell because those are its attachment proteins.